In this video, we will understand the concepts behind second messenger and their role in cellular signaling. So in every signaling pathway, there would be receptors, there would be downstream players, right? So upon binding to a ligand, a receptor would activate several intracellular enzymes. Now these enzymes, by activating many other proteins, can generate molecules known as second messengers. These second messengers can be produced numerous in number. One ligand is capable of producing thousands of second messenger. And the second messenger acts like a signal amplifier in a signaling pathway. And they convey the signal to the nucleus in an amplified manner. That's the key job of the second messenger. In this video, we would try to understand how second messengers can work like a middleman and facilitate the transduction of the cellular signal. So which second messengers we are going to look at? First, we are going to look at IP3, DAG, cyclic AMP, cyclic GMP, and in between we are also going to look at calcium. So let's begin. But biology is always fun when you learn with animations. First, we'll talk about IP3 and DAG as a second messenger. So IP3 is produced from phosphatidyl inositol 4,5-bisphosphate, which is basically a phospholipid present in the membrane. Upon cleavage, it produces IP3 and diacylglycerol or DAG. So when it is also linked with G protein coupled receptor signaling, so it's a G alpha Q signaling, where upon ligand binding, phospholipase C gets activated with the help of the G alpha Q subunit. And this phospholipase C enzyme can actually cleave membrane bound PIP2 into IP3 and DAG. Now IP3 can actually diffuse inside the cytosol, can bind to the receptors or IP3 receptors present in the endoplasmic reticulum. Now inside the cell, endoplasmic reticulum is a reservoir for calcium. So upon IP3 binding, there would be calcium outflux into the cytosol. Suddenly in the cytosol, there is a spike in calcium concentration. Calcium also acts like a second messenger, because calcium can convey the message at multiple levels in a signaling cascade. Calcium is sensed by a sensor. This sensor is known as calmodulin. It has calcium binding domain. And there are multiple kinases that are calmodulin dependent kinase known as CAM kinase can recognize the elevated level of calcium. Based on the signal intensity and the magnitude, it can convey different kind of signal. And the manifestation of this signal is by phosphorylating several downstream proteins. But what are these downstream proteins? So CAM kinase can phosphorylate metabolic regulators in a pathway, maybe a rate limiting enzyme in a metabolic pathway. It can phosphorylate cell surface receptor, might initiate or stop a signaling cascade. It can also regulate transcription factor localization into the nucleus, thereby allowing or not allowing gene transcription. So plethora of tasks can happen downstream because of the calcium level elevation in the cell. And thereby calcium, IP3, all of that work like a second messenger. Apart from calcium and IP3, another component that we missed is the DAG or diacylglycerol. Diacylglycerol, which is kind of like a lipid derivative, this is recognized by protein kinase B. So this protein kinase B can actually phosphorylate again several downstream players. That could be enzymes, metabolic regulators, etc. And that brings out the cellular functionality. Now let's talk about cyclic AMP as a second messenger molecule. So let's see how cyclic AMP can act like a second messenger. Cyclic AMP is generated from ATP, and this is a second messenger in several signaling pathway. It is underlying beta adrenergic receptor, glucagon receptor, vasopressin receptor, LH, FSH, TSH receptor, parathyroid hormone receptors, and many more. So half a dozen signaling pathway is reliant on cyclic AMP generation. Many of these hormonal pathway, which are peptide type hormones, require this kind of cyclic AMP as a second messenger. So it's very important. Cyclic AMP is generated by an enzyme known as adenylate cyclase. This adenylate cyclase can convert ATP into the cyclic AMP once activated. And the activation of the cyclic AMP, the activation of adenylate cyclase is underlying a G protein coupled receptor signaling, where the G alpha S subunit eventually gets dissociated when ligand binds to the GPCR receptor. It gets activated by exchanging GTP in place of GDP. Now it can go to the adenylate cyclase and activate that enzyme. Upon activation, adenylate cyclase produce the cyclic AMP. But what happens to the cyclic AMP once it is generated? One molecule of ligand is capable of generating several molecules of cyclic AMP. Cyclic AMP binds to specific kinases known as protein kinase A. Protein kinase A has regulatory and catalytic subunit. Regulatory are shown in red, catalytic subunits are shown in blue. So these cyclic AMP, 4 molecule of cyclic AMP can bind to 2 regulatory subunits, and this binding lead to dissociation of the catalytic subunits. These catalytic subunits are now free to diffuse in the nucleus, and they can bind and interact with CREB, which recruits CREB mediated transcription. So overall, this signaling pathway is really important in context of disease, especially in Vibrio cholerae infection. So Vibrio cholerae actually attach to specific glycoproteins and injects its toxin. It has a AB toxin that is released. Now this AB toxin in the Vibrio cholerae can actually lock the G protein in a constitutive active state. That means it would activate the adenylate cyclase for a longer duration, and as a result, lot of cyclic AMP would be generated. When the cyclic AMP concentration is so high, lot of aberrant thing also happens. Specialized channels which are cyclic AMP gated, known as CFTR, are open. Now at this point, lot of chloride gets out of the cell, and following chloride, lot of water is also expelled out of the cell. That leads to that diarrhea kind of situation in cholera. So now we understand how dysregulation of the second messenger signaling might be underlying a pathology of a disease. So how did scientists figure out that cyclic AMP could work like a second messenger? Isn't it fascinating? A molecule totally unrelated, how does it function in a cellular context to amplify the signal? So basically, this work is done by Earl Sutherland, and he got Nobel Prize in medicine for his discoveries about cyclic AMP. And he showed that how signals from one cell can be conveyed by a intermediate messenger, and he showed cyclic AMP to a crucial second messenger in the signaling cascade. Now it was the lab of Marie Curie and Gertie Curie where he used to work. So he was trying to study the role of the hormone epinephrine, and he was looking it in context of glycogenolysis. That means breaking down of glycogen in context of glucose. Now in one ingenious experiment, which is very simple, he took cytoplasmic fraction of the liver. Now obviously in the cytoplasmic fraction, the enzyme glycogen phosphorylase, which is really important for glycogenolysis, would be there. He took also the membrane fraction, which is supposed to have the adrenergic receptors, the epinephrine receptors. So in these two separated fraction, he first took the membrane fraction, and in that fraction epinephrine was added. Now obviously if something is generated after adding the epinephrine into the solution, that solution might have some diffusible molecule inside that might trigger a signaling cascade. Now this epinephrine activated membrane solution was eventually added into the cytoplasmic fraction. Note that the membrane portion was not added, only the filtrate was added. Now what he saw, and he measured the activity of this enzyme before and after adding of these filtrate of the membrane fraction. And to his surprise, he saw there is an augmentation of the phosphorylase activity. So that means something was there in that solution which has led to an increased activity of the phosphorylase. Later on, he concluded that there might be some intermediate messenger that has amplified this signal. And several years down the line, it was discovered to be cyclic AMP. Question is, in today's date, how one can check cyclic AMP levels inside a cell? There might be multiple ways. One of the way is to utilize microscopy and perform a FRET, or Foster resonance energy transfer method. There could be FRET-based sensors for cyclic AMP. Now in FRET-based sensor, there would be several excitation wavelength, and if these sensors are close to each other, there would be FRET happening. Now if the sensor domains are not close to each other, FRET would be lost. Now it turns out, upon cyclic AMP binding, these domains actually move away from each other, leading to loss of FRET. So in this case, the loss of FRET is actually helping us or informing us about the presence of cyclic AMP. This is how one can generate FRET-based cyclic AMP sensors. So these FRET measurements would be very crucial for experimental determination of cyclic AMP levels. So generally, this FRET-based sensor is based on the principle of protein kinase A. Now when the signaling is off, there would be FRET. When the signaling is on, there would be loss of FRET. And this is how one can understand the cyclic AMP signaling is active or inactive in a signaling in a cell. Cyclic AMP system is really important under several physiological contexts. It is shown that it is important for olfactory learning in fly. There are fly mutants known as rutagaba, where cyclic AMP generation was abrogated, and they have low learning index in a particular maze. In this maze, they are given to two odors. One odor comes with a shock, another odor is normal odor, and they sort of remember which odor came with a shock and avoid that odor. But fly mutant with rutagaba has a poor learning index, as if they could not remember which arm and which odor came with a shock. That clearly shows that cyclic AMP is really important underlying memory formation as well. Now enough of cyclic AMP, we are moving to cyclic GMP as a second messenger. Cyclic GMP also an unusual molecule, right? But question is how cyclic GMP is generated. Cyclic GMP is actually generated from GTP with the help of an enzyme known as guanylyl cyclase. See the resemblance with adenylyl cyclase? So cyclase AMP convert ATP or GTP into its cyclic form. Now cyclic GMP can also be broken down with the help of phosphodiesterase. Cyclic GMP is really crucial for nitric oxide cyclic GMP pathway in smooth muscles, and it is also important for phototransduction. So here is a capillary endothelial cell and a smooth muscle cell. So upon activation of a signaling cascade, phospholipase C and IP3 is generated. That lead to activation of IP3 receptors, which elevates calcium in the capillary endothelial cell. Now elevated calcium lead to conversion of L-arginine into citrulline and nitric oxide. This nitric oxide now can diffuse to the smooth muscle cell, and these nitric oxide get sort of like recognized by a nitric oxide receptor. This nitric oxide receptor eventually converts GTP into cyclic GMP by the activation of guanylyl cyclase. Now the cyclic GMP activates protein kinase like protein kinase G. Remember, cyclic AMP activates protein kinase A. This protein kinase G can now phosphorylate several downstream target. So again, one molecule of NO can activate several molecules of cyclic GMP, and that lead to a signal amplification in all these cascade. This is a common theme. One molecule of signaling ligand can generate thousands of molecules of second messengers. Now this cyclic GMP is involved in phototransduction. We are looking at the retina, where we are looking at the rod and cone cells. Inside the rod cells there are rhodopsin. Rhodopsin is basically a G protein which is activated by light. So when there is light, there are specific conformational change in these receptors. So from 11 cis retinal, it gets converted into all trans retinal. Now rhodopsin transduce the G protein coupled receptor signaling with a G protein known as transducin. Now transducin in its activated state exchange GTP with GTP. Now the GTP bound transducin can activate this enzyme cyclic AMP phosphodiesterase. So we had a mental picture that always in signaling pathway cyclic AMP or cyclic GMP has to be generated. Now let us break that stereotype and try to understand how breaking down cyclic GMP can also work like a signaling messenger, and that's what happens in underlying vision. So cyclic GMP phosphodiesterase can break down cyclic GMP into GMP. Now what does that do? In normal circumstances, when there is absence of light, lot of CNG gated channels or cyclic nucleotide gated channels are open. That allows sodium influx, which make the neurons depolarized or make them fire. But when there is light, there is enough amount of transducin activation that activates cyclic GMP phosphodiesterase. So eventually the concentration of the cyclic GMP falls down. Now when cyclic GMP falls down, the cyclic nucleotide gated channels would be now off. So no more sodium can come in through this channel and the neurons cannot be depolarized. They becomes hyperpolarized. So that leads to switching of the signal. And this works like a binary gate. In dark, there is neurotransmitter release, and in light, there is absence or reduction in the neurotransmitter release. This binary signal helps us to process the vision, because it's action potential is a all or none phenomena, and this binary message which is underlying this cyclic GMP cascade helps us to visualize things. So this is how cyclic GMP is super important in signal transduction. One thing to note, just like other theme, we can understand there is one molecule of G protein coupled receptor can eventually activate approximately 5,000 molecules of can break down 5,000 molecules of cyclic GMP. Now that tells us how the signal amplification has happened. One molecule of G protein activating hundreds of transducin molecule. Each transducin molecule can sort of activate thousands of like phosphodiesterase enzyme, and each of them can break down several molecules of cyclic GMP. So at every single hierarchy, the effect of the signaling is amplified, and that's the job of the second messenger. So I hope in this video you understood the concept of signal amplification and the role of second messenger. But anyway, I hope this video was useful. One important question that we should understand: why second messenger signaling exists? It would make the process of signal transduction very lengthy. We have also looked at other signaling regimes like nuclear receptor signaling, which is quick, right? Because it doesn't have so many middlemen. But at the same time, nuclear receptor signaling doesn't have the capacity of signal amplification. Think in a physiological context where we have to visualize very low intensity of light. In those cases, amplification of the signal would ensure greater detectability. But nuclear receptor signaling is a fast signaling which lacks the capability of signal amplification. So I hope you can appreciate the effect and the sort of like importance of importance of second messengers in any signaling cascade. So I hope this video was useful. If you like this video, give it a quick thumbs up. Don't forget to like, share and subscribe. See you in next video.